Hello, my name is Willow, and today I'll be discussing the topic of cancer genetics. We are given a scenario where in a certain species of dragon, throat cancer is one of the leading causes of death. Genetic analysis was done and 3 specific genes, X, Y, and Z below, were identified that are thought to contribute to this cancer. Before we begin, it is important to know there is typically mutations in 2 types of normal genes that cause cancer cells to develop. These 2 types are tumor suppressor genes and proto-oncogenes. Normally functioning tumor suppressor genes prevent uncontrolled growth. However, if both alleles of this gene are mutated, it disrupts cell cycle regulation. Proto-oncogenes normally promote cell division but have to be activated properly. When a mutation occurs in this type of gene, it allows uncontrolled cell division. This type of gene only requires one allele to be mutated to result in tumor formation. We can now look at the genes in this scenario and how they influence each other. If I start by looking at gene X, I can see that a family history with mutations in this gene have contributed to the susceptibility of these dragons developing cancer. I can therefore hypothesize that the offspring of these dragons are likely inheriting one defective copy or allele of gene X, and I will represent the mutated alleles in black. If dragons are inheriting one defective gene, that means they only need to mutate the other copy to result in cancer cells or a tumor. Since it's stated that benign tumors tend to have mutations in both alleles of gene X, this makes me believe that gene X is a tumor suppressor gene, because out of the 2 types of genes I described, this type requires both copies to be defective. It was stated that benign tumors tend to have mutations in both alleles of gene X, but gene Y and Z have only wild-type alleles, so they're currently functioning as they should. So this is the likely gene sequence to result in benign tumors. We can see, however, as the tumor progresses from benign to malignant, meaning it has started to invade surrounding tissue, that one copy of the Y gene is mutated. Considering it only took one allele of the gene Y to cause this tumor progression, I hypothesized gene Y to be a proto-oncogene that has now been mutated and results in uncontrolled cell division. Gene Z is still normal and functioning properly. So finally, as we progress to a metastatic tumor, which can spread and move to other parts of the body, we see that in addition to our other mutations, one allele of gene Z is now mutated. Again, since it only took a mutation in one of the gene Z alleles to move this tumor progression forward, we can hypothesize it to be a proto-oncogene that has now been mutated and cannot be activated properly. So an example like this shows how the disruption of certain gene functions can lead to the development of cancerous cells, which in turn lead to higher replication and mutation rates of other genes. The fact that these dragons had a family history of mutations in gene X led to their increased risk of throat cancer because once both alleles of gene X were mutated, the progression and mutations of the following genes took control, which in worst case scenarios led to metastatic tumors and death.